Anjali Sinha. So today I'll be dealing with physiology, continuing further with endocrine physiology. We have the next topic, parathyroid gland. So before we move on to the next, before we go deep into this topic, let's have a brief introduction. Right. So there are about 4 parathyroid glands which are situated on the anterior aspect of the neck, embedded posteriorly at the 4 poles of the thyroid gland. So suppose this is the anterior aspect of the neck and this is the thyroid gland. Okay, so they are located on the anterior aspect of the neck. This is thyroid gland, embedded posteriorly at the 4 poles of the thyroid gland. Right? There are about 4 parathyroid glands situated on the anterior aspect of the neck, embedded posteriorly at the 4 poles of the thyroid gland. The parathyroid gland, PG, I write the shortcut PG, yes. The parathyroid gland secretes 2 types of cells, the chief cells and the other one is the oxyphil cell. The chief cells, they are the predominant. The chief cells, they are predominant and are present throughout life, right? They are predominant and they are present throughout the life. They secrete parathormone or parathyroid hormone. Parathormone or parathyroid hormone. The oxyphil cells, they are less plentiful, whereas the chief cells, they are the predominant ones. Okay. The oxyphil cells are less plentiful and their function is not defined. They are not well defined. So sometimes it's said that they secrete PTH. They may secrete PTH. Okay. They may secrete PTH, but usually they do not. Usually they do not. Okay. And yes, they appear at puberty. Okay. Now, anything else? Yes. So oxyphil, these, as I told, these PTH are released at some cases. Okay. It's not for sure that they will secrete, but if they secrete, they're secreting from the granules of the oxyphil cells, and those granules are called as oxyphil granules. Okay. So the oxyphil granules will secrete the PTH. Okay. So yes, this PTH. Why are we going to study about this PTH? What is its significance? This PTH ensures, this is the major regulatory of calcium and phosphate metabolism in the body. So they ensure that the plasma calcium level is high, okay, but plasma phosphate level is low. So this is what they are doing in the body. So they are increasing the plasma calcium level and ensuring that the plasma phosphate level is being decreased. Right. So this was the brief introduction of parathyroid gland. Now moving on to the parathyroid hormone or parathormone, particularly for this hormone, which is all we are going to study in this chapter. Yes. So yes, the structure, right? The structure of parathyroid hormone. This parathyroid hormone is a peptide. It's a polypeptide. Okay, it's a polypeptide hormone. Let's write the heading as structure. Okay, it's a polypeptide hormone and it is having 84 amino acids. 84 amino acids. Okay, so this is about its structure. Now, synthesis. How are they synthesized in the body? See, you consider this as cell. Okay, which cell? Parathyroid cell. Let's take this particularly to be the chief cell. As we already discussed, chief cell and oxyphil cells. So I'm not going to discuss about the oxyphil cells because I told you that it is very rare that the oxyphil cells secrete PTH, parathyroid hormone or parathormone. Okay. So what happens is, this is nucleus. This is nucleus. Now DNA is present, right? Now this will undergo transcription and form mRNA. Okay. This mRNA is transported in the cytoplasm. This mRNA further, when it binds with ribosomes. So you've studied about this, right? Translation happens, and after that proteins are formed, isn't it? This is what we study, and we have been studying. Now these proteins that are formed are basically what do we call them as? We call them as pre-pro-PTH. We call it as pre-pro-PTH. Okay. This pre-pro-PTH is having about 115 amino acids. Okay. Now this pre-pro-PTH converts into pro-PTH. This will be transported to the Golgi apparatus. This is Golgi apparatus. Okay. So we know the function of Golgi apparatus. That Golgi apparatus functions as they'll help in package, they'll help in modification and transport, right? So first they are modifying it, they are packaging it, and then they are transporting, right? So when the pro-PTH enters or gets transported to the Golgi apparatus, these Golgi apparatus will modify this pro-PTH into PTH, right? And these PTH, they are quickly transported as transported in the granules. Okay, so we studied that they secrete vesicles, right? The Golgi apparatus. So there will be vesicles. These vesicles will be containing parathyroid hormone or parathormone. So these vesicles will have this hormone. Okay. Okay, I forgot to mention this, right. So pre-pro-parathyroid hormone will have 115 amino acid. This reduces to 90 amino acid, and this will have how many? As I told you here, 84 amino acid, right? This will have 84 amino acid. Okay, so let's read this again. So proteins are formed. You know how the proteins are formed, transcription, after that translation you have. The proteins formed are pre-pro-parathyroid hormone having 115 amino acids. Now this will further convert into pro-parathyroid hormone containing 90 amino acids. This will get transported to the Golgi apparatus. The function of the Golgi apparatus is to modify, how do they package it? They package into vesicles, right? In the form of granules, you can say. And then they transport it. So when the pro-parathyroid hormone reaches the Golgi apparatus, they are being modified to parathyroid hormone. Then they are not excreted, they are formed in the granules. We call it as the parathyroid hormone vesicles. Okay, which will have parathyroid hormone inside it. Then they are further transported. So now secreted, right? So talking about it under the topic secretion. Okay. Now, how are they secreted? The parathyroid hormone released from the chief cell. I told you this is the chief cell, right? So this parathyroid hormone, which are released by the chief cell or released from the chief cell as a result of exocytosis. Okay. So there will be exocytosis and these parathyroid hormones will be released out. Okay. Now this happens in response to the decreased plasma ionized calcium concentration. So when there is decreased plasma ionized calcium concentration in the body, this will result in exocytosis of these granules, and as a result of that, parathyroid hormone will be released. Okay, now this is sensed by calcium receptors in the parathyroid cells, right? So there are calcium receptors here present. So these calcium receptors will sense the calcium level. So they will sense that if the plasma ionized calcium level is low, then there will be that will result in exocytosis of the parathyroid granules, and the result will be that there will be release of parathyroid hormone, right? But if the plasma calcium level is high, if the plasma calcium level is high, then it diminishes the secretion, right? So this is it. Now, as I told, yes, how are they secreted? They are secreted as a result of exocytosis, okay? And this is sensed by the calcium receptors present on the parathyroid cells, which acts in response to the decreased plasma ionized calcium concentration, okay? So, regulation of this secretion. As I told, there is one factor, that is calcium, which is regulating the secretion. What are the other factors? So we have role of vitamin D, role of plasma phosphate concentration, and other factors too. So we discussed about the plasma calcium, that is secretion of PTH or the parathyroid hormone will be increased, or will be or you can say is maintained basically by the circulating levels of the ionized calcium. It will be increased when there is low plasma calcium level, and secretion is diminished or decreases when there is high plasma calcium level, right? So the next factor, regulation of secretion. So this was secretion. The next thing is regulation of secretion. The next factor is the role of vitamin D. What does vitamin D do? This vitamin D, we know, okay, I'll write the second factor. The first factor was calcium level. Okay. You can write it as plasma ionized calcium level. Okay. Next is the role of vitamin D. We know vitamin D is what? 1,25 dihydroxy cholecalciferol. This is the cholecalciferol. This vitamin D inhibits the transcription of PTH gene, okay, to form pre-pro PTH. This vitamin D will inhibit this, okay. This vitamin D, also called that 1,25 dihydroxy cholecalciferol, will inhibit the transcription of this PTH gene to form pre-pro mRNA or pre-pro PTH, and hence this will inhibit. When this is not being formed, how will this get converted into pro PTH and then PTH, and then how will the parathyroid hormone be released? It won't happen, right? So the first stage itself is blocked, or first stage itself is inhibited by vitamin D. Now the next factor is the role of plasma phosphate concentration. Phosphate. Okay. So here this is reverse of calcium. When this phosphate and when there is rise in plasma phosphate, that will stimulate the PTH secretion. The PTH secretion is stimulated when we discussed that when this is low, calcium level is low, then PTH secretion happens, right? But here, when phosphate level is low, PTH secretion doesn't happen, right? This phosphate, when it increases, when there is a rise in the phosphate concentration, then it will result in the PTH secretion. And how does this happen? This happens when phosphate, what does it do is, it lowers the plasma calcium level and inhibits the formation of this vitamin D. What does it do? It lowers the plasma calcium level, and inhibits the formation of 1,25 dihydroxy cholecalciferol. Okay. So these are the three factors. There are other factors too. The other factor we have is like example cyclic AMP, magnesium. So magnesium, if the level of magnesium is low, PTH secretion will be more. If the magnesium level is high, if the magnesium level is low, then PTH secretion will be high. But if it is high, the PTH secretion will be low. That we know. Okay. Suppose, as I told, that decreased level of this magnesium will result in secretion of PTH. But if there is severe decrease in this magnesium, then what will happen is this further or severe decrease will inhibit the PTH secretion and it will cause hypoparathyroidism. Okay, what did I tell? See, decreased magnesium concentration will confirm the PTH release or PTH secretion. But if there is severe decrease of magnesium, what will happen? PTH secretion will be inhibited. Inhibits the PTH secretion, and if there is PTH secretion low, then that will cause what? Hypoparathyroidism. Okay, this is okay, fine. So this was about it. This was the first we studied about synthesis. First we studied about the structure, then synthesis, then secretion here, then regulation of secretion. The next thing that we'll be studying about is mechanism of action of PTH. As I told, this PTH is released. Now where do they go act? They act on the target cells, and these target cells are located in bone, kidneys and intestine. That we will be studying in the next part. Okay, so before we go to the next part of the video, let me tell you about one more thing, that the normal plasma PTH level is 10 to 60 picograms per ml. Okay, and their half-life is about 10 minutes. Why 10 minutes? Because they are continuously degraded by the copper cells in the liver. So their half-life is just 10 minutes, and the normal PTH level is how much? 10 to 60 picograms per ml. So this was about it. The next part of the video will be about the mechanism of action of PTH. Okay. Okay. So we have the next part, that is mechanism of action of PTH. Now, as I told in the previous part, that the PTH, they are released. Now, once they are released, they act on the target cells, and these target cells are located in bone, kidney and intestine, as I told earlier, right? Okay, so this hormone PTH, parathormone, when released, they are sensed by the receptors present on these target cells. So basically, we've got three types of receptors. Okay, three types of receptors we have. The first receptor is PTH1R, that is the first receptor, parathyroid hormone, first receptor. The next is parathyroid hormone, second receptor. The third one is called as the type 3 receptor, that is also called as the CPTH. This is the carboxy terminal of PTH. Okay, the first type of receptor one, the type 1 receptor, is called as the parathyroid type 1 receptor. Okay, the type 2 receptor is called as parathyroid hormone 2 receptor. Then the third one is carboxy terminal of PTH. Okay, so considering this to be your target cell here, here you have the parathyroid hormone receptor. So you can also write it as PTHR. Okay, PTHR, parathyroid hormone receptor. Okay, and attached to PTH1 and PTH1 and PTH2R, there are two types of proteins. There are two types of G proteins actually. Those are GQ and GS. Okay, what are those? Those are GQ and GS. Right. Okay. Binding of this PTH. There is PTH now, as I told you, PTH is released. This is PTH. So binding of this PTH to this parathyroid hormone receptor, this activates the adenylate cyclase and phospholipase C in the membrane. So there is phospholipase C here, phospholipase C, and there is adenylate cyclase, cyclic AMP. Okay. So when the PTH binds to the parathyroid hormone receptor, which are bound to the two types of G proteins, GQ and GS, there is activation of adenylate cyclase and phospholipase C. Okay, so when there is activation of this adenylate cyclase, cyclic AMP, then it liberates cyclic AMP, or you can write it as CAMP. Okay, so this happens in the presence of ATP. This cyclic AMP, when is stimulated, when there is stimulation of the adenylate cyclase as a result of binding of PTH, then this cyclic AMP is liberated. This cyclic AMP, what does it do is it activates the protein kinase A. Okay, this is protein kinase A. This protein kinase will cause phosphorylation of, what does it do? It causes phosphorylation of the intracellular proteins. This phosphorylation of the intracellular proteins will result in the formation of phosphoproteins. Okay, these phosphoproteins now further will increase the transport of calcium and other ions. Increase transport of calcium ions and other ions. Okay, this is what this cyclic AMP liberation as a result of activation of adenylate cyclase happens. Okay, when this gets activated, cyclic AMP is released, which will activate the protein kinase A. This will cause phosphorylation of intracellular proteins. Then phosphoproteins will be formed. These phosphoproteins will increase the transportation of calcium ions and the other ions. So as I told, binding of PTH will activate adenylate cyclase as well as phospholipase phospholipase C. Right? This activation will increase IP3 and DAG. Now the question is, what does this IP3 do and DAG do? Right? This IP3 causes intracellular calcium mobilization. What does it cause? Calcium mobilization. Intracellular calcium mobilization happens due to this IP3. This DAG activates protein kinase C. Okay, this activates protein kinase C, and IP3 will cause the intracellular calcium mobilization, and this is happening when adenylate cyclase is activated. Okay, so this was about it. This is about mechanism of action of PTH. For this video, that is all, and we'll be covering the rest in the next part of the video. That is, we'll be covering that part of the video tomorrow. And so this is all for today. We'll be covering the next part of the video tomorrow, where we'll be talking about the physiological actions and a little of clinical aspects. That is going to be quite a short video, not much lengthier than this. So yeah, I hope you found this video useful, and if you did, please do drop a like and let me know in the comment section if it was helpful or if I can improve this. Yeah, so thank you so much.